I'm going to announce Chef Galvin, and thank you so much.
Thank you.
You're welcome. It's a little bit finicky.
Oh, can you guys hear me? Yeah, I guess I can hear myself. Thank you for that very nice introduction, Tita, and it's a pleasure to get a chance to talk to all of you about a bunch of things that are near and dear to my heart. Let me start with Parenteral Drug Association, PDA. One of the things that's interesting about this is how many people in the room already knew when they came in here what parenteral meant? Okay, quite a few, which is amazing. Of course, when I first saw it, I was like, parenteral, is it something that the descendants of it are the drugs? Is it drugs that are exclusively for older people that may be parents, whatever? I got to say that this was an education for me. And I realized that this is the amazing thing, is this is the future of drugs. Yeah, I think it's the future of drugs because I believe that gene and cell therapy is the future of drugs. And I think gene and cell therapies aren't going to be pills, at least for a while. They're going to be infusions. They're going to be shots. They're going to be cell therapies where we pull cells out of you and then reinfuse them back in. So the Parenteral Drug Association, PDA, is becoming more and more relevant every single day. And so it's my pleasure to be able to support them and come here and speak at this event, and I hope there'll be many more of them. It's really two things that are very near and dear to my heart is the whole idea of bringing solutions to patients, and then the second thing being just gene and cell therapy in general. I think that gene and cell therapy is nothing less than a revolution in pharmaceuticals. I think that everything that you accept to be true or that the industry accepts to be true about pharmaceuticals is about to just all fall apart. It reminds me very much of my days in other types of technologies like microcomputers. When I was in microcomputers back at Apple, the companies that dominated the computing industry that Apple was going into were, and see how many of these you actually have heard the name of before, Burroughs, Sperry Univac, the GE mainframe division, Control Data. You've probably heard of IBM. You know why you've heard of IBM? Because one visionary leader of IBM looked at microcomputers and he said, hey boys, we're screwed. We got to figure out what we're going to do after the revolution. And guess what he decided to do? He said, IBM means business. We can install PCs for major businesses. That's a business. Our brand is worthwhile in the new world. I think you're going to see the same thing in the pharmaceutical companies. I know you can't imagine the day where there's an audience like this and people are like, remember Roche? Remember Pfizer? Remember Merck? You heard of those, any of those companies? Right? Why is that? Well, they've been so successful that they're literally calcified in an old technology. And what I'm going to show you today is a new technology that completely changes what the possibilities are. And once the market, patients, get exposed to this, they will accept nothing less. It makes the word cure no longer a four-letter word. We're not allowed to say the word cure because for 30 or 40 or 50 or 60 or how many years, cure has not been part of drugs, right? What do all these companies do right now? They offer you stuff that treat things. That's at the best case. In the worst case, they offer you things that offer you hope that you may not die as fast or as painfully. And that's not going to work if cure is no longer a four-letter word, if cures are actually possible. And they are. So let me ask you a question just to make sure you're all still listening. Let's say you go down to the doctor because of whatever symptom and they do some imaging and whatever and they come back to you and they say, got some bad news, you have cancer. If you're at that point, would you rather have surgery, radiation, and chemotherapy, or would you rather they said to you, you're going to need a shot for that, and then they gave you the shot, and they said, okay, go back to work, you'll be fine in 6 weeks. Not much of a choice, right? I think this is actually possible. I'm going to show you how we're shooting for that. I think we may actually make progress towards that. This was the dream of AGT when it was founded, was to send radiation and chemotherapy the way of bloodletting and leeches. And you can understand why you can dream about something like that, or why you would, sort of think about that as an aspiring drug developer, right? I know there's a future. I don't know how soon. I think it may be sooner than we think, and I hope I'll convince you of that today, where you'll be telling your children or your grandchildren, you won't believe what we used to do for cancer. We used to beam ionizing cancer-causing radiation through your body to treat it. We literally were using things that cause cancer to try to treat cancer. Then, if that wasn't effective enough, listen to this. We poisoned you as much as we possibly could without killing you, because we had certain substances that would poison cancer just a teeny bit more effectively. And these oncologists, they were like magicians. They would walk this thin line between killing you, they didn't want to do that, but they had to get past this level where the cancer got killed and you somehow recovered and the cancer didn't, and we called that a cure if it lasted for 5 years. And the kids will be like, wow, I can't believe it, right? Yeah, what's that?
Barbarians.
Exactly, yeah, that is barbaric. Yeah, like back in the days when wars weren't fought in virtual reality. The kids are going to be seeing a lot of different things than us. Yeah, I think that is a reasonable dream for this whole room. I think it's a reasonable dream for the whole world that we can make that kind of progress. And I think where it's going to be made is in gene and cell therapy. So let me start off with a little bit of contextual information so you can understand how I got into this thing. So as you may have gleaned from my talk of Apple computers, I used to be in the computer industry. In fact, it goes back further than that. If you can imagine me in the 7th grade, so that's about 2 or 3 million years ago. And I see a teletype, which is what used to attach to mini computers, which by the way are all gone as well. Who's heard of Digital Equipment Corporation? Anybody heard of that? Wang? Okay, there you go. Heard of Apple? Microsoft? Okay, keep that thought in your head. So, I was interested in, what is this new thing, computers? And I got an independent study and I learned how to program, and I absolutely fell in love with computers. And why? I wasn't in love with programming. I was in love with what I could see the future of computers were. That they were going to eliminate all the most boring rote activities from our lives. We were never going to have to add numbers together anymore. We were never going to have to format documents. And then in the 7th grade, I had the privilege of wandering around MIT on weekends because I lived nearby and my dad was graduated from there, so I felt comfortable on campus. And I was just looking around for free computers to use. And I saw robotics and sensor technology, and then I realized, oh, it's not just going to format numbers and documents. This is going to be building all our stuff, driving our cars, flying our planes, doing everything in I could see that this trend in technology. I knew that these giant computers that nobody could afford were now in my junior high school, what you would call middle school today. And, they'd come down from room size to, room size. I mean, this whole, area here could be one computer for a Fortune 500 company, to being something that you could fit maybe back in that little alcove area back there. That was mini computers. And now, of course, we're carrying things that are 1,000 times more powerful in our pocket. So the trend was really obvious. Better, smaller, cheaper, right? So if the capability of something is going up constantly and the cost of it's going down, what's possible? And the answer is anything. If you can dream it, you can do it. That's the nature of technology in my mind. That's what I fell in love with computers. And I saw it over and over again because after that I was teaching at MIT, I was teaching at Harvard. After I graduated Harvard, I got a job offer at Hewlett-Packard. I got out there. They were really interested in software and computers, but they weren't interested in what they could do for people. So I didn't really actually enjoy it there that much. But my next gig was Apple, and they got it. You know, one thing you can say for Steve Jobs is he understood what computers could do for people, and he was interested in getting it there. That's something that not anybody else in the industry was interested in. Computing was computing, right? And Steve Jobs said, no, computers should be like an appliance in your life that enhances your life. And he wanted them to be approachable. He wanted them to be easy to use. This was very, very important. That really appealed to me because it wasn't about the software. Anybody could write the software. It was about what the computer could do. So there I got my street MBA, and I learned about evangelism, which you might even realize I'm doing here today, right? If you've got something better, go out there and tell everybody about it. If people get it, they'll actually power the revolution. So everybody in this room, you're now part of the revolution, assuming that after you see this presentation, you're convinced that a new technology has come to town. And I think I'll convince you of that. So I did really well in Silicon Valley. After Apple, I did a bunch of startups. I had some successes. I had some ones that failed but didn't kill me. I invested in a lot of technologies because I had a good eye for it. And I look at my bank account at about age 41, and I realize I don't have to work anymore. I can retire. And I'd been working really hard. And so I thought, retirement, that early retirement, that's what everybody dreams of, and I retired. I bought a house in Hawaii. I was flying back and forth between my home in Silicon Valley and my home across the street from the beach in Maui. And that was fantastic for about 5 years until it got really boring. And I thought, I'll dabble again in technology. I thought I would go back into computers because that would make sense, right? Sit on a board, make a small investment, mentor some people. And as luck would have it, people started hearing that I had money to invest. And of course, then you get business plans from all over the place. And one of those business plans came from National Institutes of Health. It was a postdoc in the lab of somebody that I later found out was exceedingly famous, a guy named Roscoe Brady. Dr. Roscoe Brady is famous for having discovered the root causes of Gaucher's disease, which is a horrible disease. If you have it, you can't metabolize lipids. The fat molecules in your bloodstream get into your organs, they swell, your organs burst, and you die. And it was nearly 100% fatal by the time you were age 20. And in your teens, you were like this. And he realized all it is just one broken gene. You're missing one enzyme in your bloodstream. The technology for enzyme replacement, or for making enzymes for your body, was getting a lot of momentum at Genentech. And he thought, you don't just have to make human insulin in these cells. We could take these same bioreactors and make the one missing enzyme and then put it in people's bloodstream, and that should make the disease go away. So he's one of the pioneers of enzyme replacement, and it worked. And NIH gave it away for free to Genzyme, who started selling them $1 billion a year, saving people's lives, right? Which is a good thing for everybody. I'm sure the Genzyme stockholders loved that. But I think NIH was quite happy about that as well, because that was their job, was to break new ground and to make new technologies that would end human suffering and premature deaths from these diseases. So Roscoe Brady doesn't even have a PhD, but he became a researcher because he had these patients, and then he, with his passion for actually solving problems for these people and his access to NIH, he just went through this whole process and did something amazing. Well, he'd done all that stuff before I met him. The other thing he'd done is he thought, how can we improve this? Right, think about that. He solved Gaucher's disease. He was saving lives every single day. And he was like, how can I save them better? And his answer was, instead of having to get shots every single day of the missing enzyme, what if the broken gene could be fixed, replaced? Then, essentially, you would be the medicine. We'd fix it once. You'd be making your own enzyme. And by virtue of that, you'd be normal. Sounds like a cure, doesn't it? And the way he decided to do it was with viral vectors that were kind of new on the scene. Viral vectors essentially are repurposing viruses to carry genetic cargoes. But it's not really repurposing them to carry a genetic cargo, because they do that already. They just carry a malevolent cargo. But it turns out that around that time, scientists had discovered reliable ways to crack open viruses, scoop out the viral DNA, throw it away, and replace it with whatever they wanted to. And he actually decided that the way to do this was to take HIV, because HIV had a unique quality that it literally installs genes permanently in its target cell, in the cell it infects. And he said, you know, for these sort of diseases, we want it to last forever. We want to modify cells so that for the rest of their life, they're making the enzyme that you need. So he started cracking open HIV, scooping out AIDS and throwing it away. And what we call what's left over, this empty shell that can carry anything, sort of an empty stealth bomber that has the delivery capability but nothing inside, we call it lentivirus. Actually, HIV is a lentivirus. Lenti means slow. HIV is a slow virus. There's a class of these things. So, but when you haul it out, we now call these lentiviral vectors or lentiviruses. That was his concept. And he had the cutting edge technology, and then he was getting into his mid-70s, and it was retirement time, and there was some politics at NIH because of, it wasn't anything having to do with his lab, but they passed some new rules that said you can't be the head of a lab and be a consultant for a company anymore. And so he had to choose between staying a consultant for Genzyme, and you can imagine how that pays, right? And you're 78 years old, right? Or keeping his lab, and so he said, I'll retire. And they didn't want to see him go, so they made him a scientist emeritus there. So he always had an office, he always had an assistant, he always had a conference room, but he wasn't allowed to run a lab. And his picture's still hanging in Building 10, and unfortunately he passed away in 2016. But anyway, so what was, back when I met Roscoe Brady in 2007, what were they going to do with all his work? They were going to shelve it, basically throw it away. This was right at the time I met him. I mean, talk about serendipity. I'm talking to him, and he's explaining to me about viruses, and I'm remembering my history in computers. I'm remembering my 10th grade biology about DNA. And right at the first meeting, it just struck me. I said, I think you're about to throw away the most important and biggest and most valuable thing I've ever seen in my life, and I've seen computers. And he was excited about that. And so I said, could I just take this stuff? And he goes, I'll check with legal. And you know what legal said? They were like, sure. It was like, whoa, I mean, this is just crazy. They hadn't patented it. They were like, they didn't know where it was going to go after that. NIH is really concerned about keep moving it forward. And so I hired some of his postdocs, and AGT was born. And so, you know, this technology is born out of really personality attributes of Roscoe Brady in some ways, and some just synergistic background that I have. The attributes of Dr. Brady were really, really simple. He never cared about money. What he cared about was people, patients. When he saw people in his medical practice who were dying of this horrible disease, he just thought about it. He had passion for them. He had compassion, and he had passion for finding a solution. And he figured it out. That's what smart, motivated people do. Like everybody in this room, you don't even realize what your capacity is for greatness. But if you take your intelligence and you find something that you're passionate about, something that you really love, then it isn't a 9-to-5 anymore. And a lot of the great ideas that I've come up with for AGT have happened in the shower, my quiet time. The one place where my wife won't find me and give me another item for the honey-do list. So you know, AGT is sort of that way too. I saw an opportunity. I knew this would be big one day. Why? Because I'd seen computers, I'd seen software, I'd seen the internet, I'd seen apps, I'd seen IT. I knew what this technology was going to do. I knew it would eventually be hugely valuable. I also understood something that a lot of people in drug development don't understand. And that is that if you're looking at the software of the human computer, DNA, and you want to be the software programmer that's going to go in there and make the most difference for the most people in terms of reducing suffering and reducing death from serious human diseases, you need to find efficiencies. It's not just about finding a solution. It's about a whole industry now of software, a new type of software. If you want to do something really special in this software industry, which I think is just beginning and that will last for at least 100 years, that really is about discovering ways that we can make small tweaks in DNA that can make a difference in systems biology to interrupt the drivers of disease or to mitigate diseases in other ways. If you wanted to, do that right, you'd look back at the last software industry and you'd say, who did it right back then? Who did it right? Microsoft and Apple. And trust me, back then, all we heard was how crazy we were. The executives of Apple, after the Mac came out, they went and they demonstrated it to the #1 industry analyst. And you know what he said to them? So they're showing off their baby. They're so proud of this, right? Look at this, the graphic user interface. You can point to things. You can move them around the screen. You double click, it opens up. You can type into it. Anybody could use this thing. Your grandmother could learn it. And he said, yeah, that's all fine and nice as maybe a toy or a game or something. But that's not how real computer people use computers. He said, here's how they use it. And he leaned back in his chair, and he pulled that keyboard, which used to be attached by like a phone cord, that curly Q cord, into his lap, and he put his feet up on the desk. And he goes, this is how they use it. And they're not going to want to keep leaning forward and moving this stupid little mouse thing. They sit back here, and that's the efficient way to talk to a computer. I mean, how wrong could he be, right? So, it's not a... There are a huge number of lessons from the microcomputer industry that are really relevant to the future of pharmaceuticals. And who are the people that can't learn them? Pharmaceutical corporations. The nail that sticks up gets hammered down, right? That it is, nobody in a pharmaceutical company gets anywhere by saying, we're nuts. We need to go that way that's totally different than everything we've been doing for the last 30 years in this multi-billion dollar company, right? It's a great way to be shown the door, not the executive suite. So they can't change. It's sort of in their DNA. The same way that Burroughs couldn't change, GE couldn't change. That's one of the things I call it the consequences of hubris, and the hubris that comes from success is really, in some ways, the most cemented hubris. These people are truly better than me. They're truly better because they've proven it. They run billion-dollar corporations already, just like IBM was a billion-dollar corporation when Apple was in the garage. Okay, so I don't think it's game over the way they think it is. I don't even think they think it's game over anymore now that they're starting to buy up this technology that you've probably seen. So that is the arc, right? It comes from my background in technology. I just fell in love with this. This is so much more important to me than even computers. This doesn't just relieve us of the rote activities of formatting documents. That's been done. This is going to relieve us from suffering. It's going to relieve us from the biggest threats in life. Are you more likely to get cancer or to be involved in a terrorist attack? You're like 90,000 times more likely to get cancer, right? This is the real security issue. These are the real threats in our life. We're working on something that is just, for me, it's all just about love. It's about actually appreciating all the people around you and actually it feeling fulfilling if you can do something positive for them. And computers were that way, but this thing is computers on steroids. This is going to do so much more because if you could, have a fancy car or you could feel really good, really healthy, and be mobile and, and live, which would you choose, right? Your health really is the most important thing, and the only reason we've lost sight of that is because the industry's only been able to do so much for health. But we can do much more with this technology. Can you believe that's slide 1? I got 10 slides, so we'll be here about 4 days. I hope you're ready for it. I'll move more quickly through the other slides. So let me just go ahead and make the point here. The human cell is the organic computer, okay? A regular computer processes zeros and ones. Has everybody heard that? It doesn't matter that it's showing you something on a screen. A screen is just what's called a to D or D to A conversion, analog to digital or digital to analog. Sensors are the same way. Once it gets inside your phone, it's just zeros and ones flying around that are represented by circuits that are either on or off. It's called binary, right? And what does it output? Zeros and ones. That's all that computer can do. And if you attach it up to clever things, you can start to do more things like servo motors could turn your steering wheel, right? A sensor could get the temperature in this room and decide to open up a vent or turn on the heat or whatever. That's what computers do. And your cell, though, is the most sophisticated computer that you can imagine. First of all, it's not zeros and ones, it's ACTG, the nucleotides of your DNA, right? It's in base 4. That's one level of sophistication above the computer already. But here's where it really is something. It doesn't output just more ACTGs. That's part of what it does, because inside your body, you're actually producing RNA and stuff like that. But the net effect of the human computer is enzymes and proteins, the building blocks of the computer itself. If you want to get an immediate sense for how much better the organic computer is than the digital computer, remember that the organic computer comes with a command that says, make a copy of yourself. It's called mitosis. I mean, that would be great if you need a new cell phone for your kid or something and you could press a button on your cell phone and it could do that, right? So now the parallels keep going. Base 2, base 4. Software. Software is the order of the zeros and ones. Every command in a computer just boils down to the order of the zeros and ones. And it recognizes that pattern and it executes a command. It might be to add 2 numbers or move a number from here to there or compare 2 numbers or something. Computer commands are actually quite simple. With the software in your body, the commands are genes, and genes can be any length. In a computer, they're fixed length words. They used to be 8 bits, then they went to 16 bits, then they went to 32, then 64. They're usually some multiple of 8. But it's whatever computer you're using, it's one of those, and every single command is the same length. In genes, any length. You have short genes, long genes, but it's all the same. The order of the ACTGs decides what the cell does. Your operating system on your computer might be Windows, Apple's iOS, Android, whatever, right? But in your body, that operating system is your DNA. It's everything that the cell is capable of doing is all packed into that operating system. Just like everything that your phone is capable of doing is in the software of that phone. Does it mean that the phone's always doing the same thing? No, it's doing whatever you tell it to do, right? So my phone isn't doing the same thing as your phone or your phone right now. It's we, as the operators of our computers, decide what the computer does. In DNA, the cell has a manager function. The cell itself knows what the cell is supposed to do, and so it knows what part of the operating system to run. That's why all your cells can be different, even though your DNA is the same. So it's got a built-in operator, which is also pretty amazing. You are just stacks of trillions of collaborating organic computers that make up everything in you, even your thoughts, right? Even your ability to hold memories, your feelings, your muscles, everything is made out of collaborating computers that they communicate with one another in order to get very complex functions, right? You wouldn't be much good. I mean, bacteria is all the same. But once you differentiate, right, the cells work together to make really sophisticated functions. And they communicate not just with electricity, like your personal computer, right? Your Ethernet is just electric pulses going over a wire. No, they can communicate electrically. You know about that, nerves and things like that. Chemicals, many different types of chemicals. They literally can release a hormone that changes your whole body. Or they can release cytokines that just move a message from one cell to another. And they can even physically bump each other, and there's probably other ways that they communicate that we haven't even discovered yet. And then here's the big one. The digital computer comes with a manual. The human computer didn't come with one. That's what makes it such a challenge. And also what makes it so exciting is that we're discovering the functions right now, and there's been an explosion in that kind of discovery. Why? Well, it may have been sort of a turning point, the Human Genome Project, because it dropped the cost of experimenting with the human genome at the same time as giving this catalog. But the main thing was we put $6 billion in 2 projects into just getting one sequence of one person's genome. And in doing that, it was like a moonshot where all this technology fell out and all this investment turned into huge competition for the reagents, the materials, and the equipment to do that kind of work. And then suddenly, any university lab could have a PCR machine. Any university lab could go ahead and order a viral vector and knock out a gene and see what happened. And so what happened there was we started to discover how the body works, right? Because we have genetics, we have systems biology, we know a lot about systems biology, but now we can make little alterations in pathways within cells and see what happens. And so it's like the black box, we have a way to sort of shoot little test rays into the black box and see how it reflects. And we're learning so much more about, the manual for the human computer. We're getting the list of commands. We're figuring out what those commands do. We understand how genetics tie with those commands in your body. So we know how your immune system works, and we know what genes control your immune system. We know how cancer moves around your body. We know what genes are involved in that. And then we have the last part of the puzzle in this 3-planet collision that's going on, and that is gene editing. Right? I already said viral vectors, right? Well, now with these viral vectors, they're getting better and better and better and more reliable. We're even coming up with editing technologies like CRISPR. And what that says is that even that part of this whole equation, we're not just learning way more about how the body works. We're not just learning way more about how genes impact how the body works, but we're getting tools that are also on the rise in terms of, the capabilities of knocking out a gene and testing for something. And at the same time, the cost is going down like a rock. What's going to happen in that situation? What could come out of that? Anything. If you can dream it, you can do it. We're at the beginning of the technology curve in pharmaceuticals. High tech has finally come to drug development. We no longer have to discover drugs. We can design drugs from the bottom up because we now have tools that can alter the body in very, very specific ways. And how is that? Well, the cell, I'm going to really go through this quickly, is basically a factory, right? It makes enzymes and proteins. The manager, I told you, decides what part of the DNA inside the nucleus, and he sends out commands all day long to the machinists. That's called messenger RNA. And machinists are really dumb. They just pick up the nearest instruction and they make whatever it says. It's like a recipe for this enzyme or something like that, right? And so viruses evolved alongside of us. They've been around for 1.5 billion years, roughly. And they have found a clever way to trick the cell into doing something else. Did you know that viruses on computers are actually named after those viruses? I hope so. That's why we call them viruses. They hijack our computer, and viruses hijack cells. They come over, and what do? They just drop a bunch of messages into the cell. And the machinists, like I told you, they're dumb. They don't know which ones are real from the nucleus and which ones are from the virus. So they just start executing the nearest command. What are all those commands? Make copies of me. This is a very sophisticated thing that they're doing, right? They were making single enzymes or whatever. Viruses literally can drop a few genes in, and they'll repackage a very sophisticated virus that has a shell, a protein coat, it has a vector inside, and a packaging signal to pull them together. I mean, these things are really sophisticated stuff. It's not just like making one thing. They're making three things that work together and make viral particles. The cell gets so hijacked by this that it uses every single piece of material in the cell. Remember, the cell is working.
Disconnected. Bluetooth connected.
Okay, all right. I thought the rapture was on us. I was looking to see who was going to disappear and just be a pile of clothes. Okay, we're all still here? Good, all right. So the cells are working in the materials they're made out of, and what do? They start pulling things from the walls, the ceiling, the floor, the machinery, everything just starts becoming viral, these viruses, and finally the cell lyses, the virus escapes, and they go on and they infect other cells, right? That's a very sophisticated thing that viruses are doing. Now that's what I call a smash and grab virus. Why? Because as soon as the cell has that kind of virus in it, it's a goner, right? Because it's going to spend all of its time making virus, and it's going to exhaust the cell so quickly that the cell will explode or implode or lyse, and then they'll go on and infect other cells, smash and grab there too. I mean, they destroy everything they hijack. But there's a more clever virus called an integrating virus. In this case, I'm using HIV as an example, that I like to think of as the 007 super spy of viruses. So instead of just dropping a lot of instructions on the floor of the shop, it sneaks across the shop floor, it breaks into the manager's office, it picks the lock on the DNA safe, and it just throws in a few new recipes, a few new commands. And it's even smart enough to tag those commands so that the manager thinks those commands are important in his shop, in his cell. It starts executing them alongside all the normal things, right? So it's making normal messages for the cell off of the DNA like it always does, and then every once in a while it'll kick out an instruction to do what? Make another HIV viron. This is why they're called lentiviruses, because that's a really slow process. The fast process would be to completely hijack the cell. The slow process is the way that HIV does it, but it's got a really neat attribute, and that is permanent edit, basically, to your genome. Now, it can only insert a gene, right, or genes, but genes that you can insert can actually turn off other genes. So it has rudimentary editing capabilities. You can put something in there that you need, that you're missing. You have a monogenic disease like Gaucher's disease. We can take blood cells out of your body. We can take a lentiviral vector carrying the glucocerebrosibase gene. We can infect them, and for the rest of their life, they will make glucocerebrosibase. We add that enzyme into that cell. But we can also put in things there that are synthetic genes that make interrupting RNAs that can shut off whatever gene we design them to shut off. So we can re-regulate any gene. It's not now only that we can add things. We can essentially add things that subtract things. And so we can readjust the expression of your genes in a positive or negative way. And that's exactly what we do. So we hijack the hijacker. We take HIV, we crack it open, we scoop out AIDS, we throw it away, and now we have that empty stealth bomber I was telling you about. Then we figure out what gene will make you feel better, will replace the thing that you're missing, and we can just insert it in that capsule, or we can insert things that will shut off other genes. We can stick it in there. As a matter of fact, we can put 3 of these constructs in one lentiviral vector, repackage it up. We remove the replication capability of this because we don't want to accidentally create a new virus. So none of these, what are called viral-like particles, have any capability of reproducing themselves. That's a very important safety feature. So we end up having to duplicate these for as many as you need. So if we wanted to treat your whole body and you have 3 trillion cells, it's a lot of these particles, nanoparticles carrying one dose with a maximum capability of installing that gene into one cell. And in fact, it's normal that you have to have a 2 to 5 MOI to get sufficient number of cells to get therapeutic expression of your new, what's called transgene. Once it's packaged up, it looks a lot like the HIV thing. You got a normal operating cell, but hey, look, this one has HIV in it, right? So let's just hold that thought for a moment. So this new lentivirus comes along. It's HIV, but it's no longer dangerous, right? It's carrying a therapeutic gene for you. And it attaches those genes to the DNA, and they start expressing. Of course, if it's an add gene, the plus sign there, it makes a new enzyme or protein. If it's a minus gene, it will send out something that will attach to the message from HIV or anything else that we want to shut down, any virus really, and destroy that message. Once you attach that siRNA to it, all it has to do is match its 19 to 21 base pair, those ACTGs. It's called hybridization. Once it sticks on, the machinery can't interpret that messenger RNA anymore. And as a result, it recycles in the cell. So it's like shutting down the gene. You're not editing it out. Eventually with CRISPR, we'll literally edit out things we don't want anymore. But that's not reliable enough for the clinic, and we can talk about that in the Q&A if you're interested. But this is really reliable. We can shut down things that we don't want, and we can add things that we want. And then here is something really important. We have, in this type of software, an if-then statement. And the if-then statement is called a specific promoter. It's like a smart fuse. It can measure something inside the cell, and from that thing, it'll look at any enzyme, any protein, and from the enzyme or protein, you can actually figure out what type of cell you're in, or whether the cell has a particular disease condition. I'll give you an example. If I only want to treat your liver, I could put lentiviral vector into your liver, but I'll put an albumin-specific promoter on the front of each one of these genetic constructs. And what it does is it only turns on the genetic construct. It only turns on the drug if that condition is matched. So if there's no albumin, which would be very uncommon anywhere but the liver, then the gene never turns on, right? So it's this safety switch that does what? It shields the off-target tissue, the tissue that you didn't want to treat, from the treatment. Why do most chemotherapeutics that fail? Because you put them in your body, they have no idea where to go. They treat every single cell the same way. They may do something really important and positive for the cells that need the treatment, but frequently the off-target effect is the killer. And so that it turns out that the product never makes it to the market. Well, now what we can do is we can guarantee it only goes where we want it. Now, what's an example of a disease state? What if you have prostate specific antigen in one of the cells, a high level of that in one of the cells in your prostate? It's probably got prostate cancer. So we could treat your whole prostate, but we can drive it with a prostate-specific antigen specific promoter, and it will only turn on the drug if the cell has the cancer. So we can put in something that's really powerful and deadly. Why? Because we know it won't turn on unless that cell is showing a sign of the disease. That's not actually how we're curing cancer. I'm going to show you how we're going to do it in just a minute, but it shows you the basic idea, right? It's what gene and cell therapy is bringing. It's not just the ability to go down to the absolute basic drivers of every single thing that's going on in your body and re-regulate that DNA, that operating system, that software that's running you. It's that it actually, you can combine logic and you can target this stuff down very, very narrowly, very specifically, and you can eliminate all the off-target effects, or most of them. And so what's really important about this revolution is, one, power, because we're down there at the root drivers of everything. And number two is the ability to ratchet up that power, to direct it specifically to where it's needed, and to isolate the rest of your cells from off-target effects, and that will allow us to get this thing through the clinic. A very powerful drug, but only acts where it's needed. Unlike anything else that you've ever taken. Aspirin goes everywhere, right? Monoclonal antibodies go everywhere. They do have a little bit of tropism to specific receptors on cells, right? But you know something? We can actually take that tropism, we can put antibodies on the outside of a viral envelope and even make the virus tropic to certain things. So that's another way that we can target it. But this is going to get better and better over time. Remember, the technology just keeps getting better and keeps getting cheaper. So I can imagine a day where you can target it so specifically that you can predict every single drug in your system. You know exactly what it's going to do. We're not there yet, but we will get there. So getting back to Roscoe Brady and my initial meeting with him, when he explained viral vectors to me and I understood this, what I just told you, my head practically exploded. I didn't want to sit on a board. I didn't want to mentor anybody. I wanted to cure some diseases. And I told him that day, I said, you know, blind people will see again. Cripples are going to get out of their wheelchairs and walk. The lepers will be healed. This is the stuff of miracles. He was really excited about that. And I said, you know, somebody needs to be the Microsoft of this industry. That he didn't get. But maybe some of you are actually getting that right now. Because again, this is the software revolution for the next 100 years. If you were in the 1960s and got a programming job with digital computers, right? Then you would have had to work for IBM. That was the only one around. Well, today, assuming that you were still mobile and interested in working and hadn't retired because it was a great paying job being a software programmer all that time, you could still get work because that revolution is still on. It's not done yet. Obvious. It's better to do that than enzyme replacement. You don't need shots every day. If you can do a one and done, just replace the gene where it's needed, right? There's 7,000 of those diseases. They're rare. What does a rare disease mean? I think it's what, under 20,000 a year? I don't know. Anyway, what's that? 200, okay. Small diseases, right? But if you add up all 7,000 of them, 1 in 10 people has a rare disease. And you add up all the money that's spent keeping those people comfortable, right? It's not curing the disease, just keeping them comfortable. $2 to $4 trillion a year. The whole pharmaceutical market is only about $1 trillion. This is why Goldman Sachs, has anybody heard of that company? That's why they predicted that this industry is $4 to $5 trillion. And I think they estimated low. You know why? Because I can tell you where you get $3 or $4 trillion of that already from rare disease. They haven't even gotten to cancer, infectious disease, all that stuff. And they haven't gotten to cosmetic use, right? Once again, you might trade in your Lamborghini for having the body and skin and hair and muscle tone of a 20-year-old for the rest of your life. Right? Because it truly is better to feel good than to look good. So stepping out of the Lamborghini doesn't do as much for you as actually feeling good. And so there's a lot, the whole healthcare industry today is $10.5 trillion, including those pharmaceuticals. So a lot of it's going to come out of the treatment stuff and go into the pharmaceuticals, and the pharmaceuticals are going to find our way into life improvement stuff. And remember, why is it that pharmaceuticals haven't grown to more than $1 trillion? Certainly people want solutions. It's that the industry can't deliver them with those old technologies, right? We're paying as much as we can afford for the healthcare that we're getting. As a matter of fact, it's breaking economies in some places. But if instead of knocking back your cancer, like let's take Avastin as an example. I love beating up on this one. So this is a Roche drug. It's a monoclonal antibody. Sells $7 billion a year. If you have a terminal cancer for which it is prescribed, your doctor will offer it to you and your insurance company will pay for it. It'll cost somebody, let's call it society, but your insurance company, $125,000. Who wants to venture a guess? What, if you compare somebody that takes it versus somebody that doesn't take it, what's the extra overall survival for the person that takes it? How much extra time do they get for $125,000? Any guesses?
6 months.
Yeah, see, these are all good guesses, right? The answer is 6 weeks. And there's no guarantees of that. That is just statistically from the phase 3 trials. That's it. The reality of it is that they're selling you nothing but hope. And you can't afford to pay an infinite amount of money for hope. But what if we made a drug that knock back your cancer for 5 years. Even the US government says your life is worth $100,000 a year in productivity. And that doesn't even include just your joy of living and being with your family and all that stuff. This is just your hardcore productivity in society. If a drug puts you back on the street for 5 years, that's $500,000 worth of value. And remember, you don't need to spend all the money on dying, because if you die of those diseases that you have asked, it usually costs the insurance company somewhere between $300,000 and $500,000 for you to die. You get nothing. But if it could be a shot, like I was talking to you about before, and then you walk out and you go back to normal life, paying taxes, paying your insurance premiums, enjoying life, working, producing stuff. Okay, now that is worth a lot. If we do that for $500,000, that's a break even. Why do you buy a computer? Because it looks good? Because you can't live without it? No, because it's an investment, right? It actually makes you more than it costs you. Drugs are going to be like that in the future. That thing that extends your life for 5 to 10 years is going to cost $300,000. And it's going to be a bargain. It's going to be an investment in your future, not a cost to the healthcare system. It literally is going to give that more value than it costs. So let's talk about cancer cure. My point here with this slide is that this is already happening. I'm not talking to you about science fiction. Okay, in the 12 years that I've been in this business, the drip, drip, drip of these drugs has started. So we have a cure for blindness. Let's turn it. You have Leber's congenital amaurosis. Instead of being blind at 15, you can see. How much is that worth? It'll cost you $850,000, but I think we can do it cheaper in the future. By the way, what else is cool about software? It's super competitive. The first solution is not always the best solution or the last solution. You're not using Visitel for your spreadsheets, are you? Or Lotus 123. No, you're using Microsoft Excel because they pack way more functionality into it for the same price. Right? So all these prices come down. It's very competitive. Spinal muscular atrophy, if you were born with that, 1 in 5,000 boys in the United States, if you die in a respirator never having learned to sit up straight or hold your neck straight or roll over, what kind of life is that? Well, now they go ahead and they put in $1.5 million worth of viral vector for a total cost of $2.1 million. Right? That's a lot. It sounds like a lot of money at first. Right? Guess what? You learn you're okay. You have a life. You go from no life to having a life. Your parents go from suffering, watching you, to seeing a normal kid. I mean, how much is that worth? Whatever we can afford. It's priceless. Here's the thing that people keep neglecting. How much does it cost you to die? Babies born with SMA don't just curl up and disappear. No, they need a huge amount of medical attention. They keep their parents at home taking care of them instead of being at work. The cost of that baby was way more than $2.1 million. Now he's got a life. I would argue that his life was pretty much free to society. That $2.1 million was a bargain. ACA skid, bubble boy disease. Turns out it's one broken gene in your bone marrow. They fixed it. There's a new one, X-linked skid. Fixed. Can we cure HIV and other infectious diseases? You bet. And this is an interesting one. There's 2 different types of cancers. And I should say AML down there instead of ALL. But acute myeloid leukemia, acute lymphoid leukemia are these deadly diagnoses of a late-stage leukemia that you cannot cure anymore. So what does it cost to cure it now? Under $500,000. Have you guys heard of NICE? It's the insurance company for all of the UK. The head of NICE said those 2 solutions would be worth it at twice the price. Now, why would he say something crazy like that? Because he's right. Because again, people with acute myeloid leukemia don't just disappear or evaporate one day. They go to the hospital and it costs about $1 million while they die, and they always die. Now, for $500,000, they're going back to work. They're paying taxes. It's a bargain. And that's the future of all these things. So don't worry about the cost in this thing. Now dream with me for a minute about the shot that cures you of cancer. So how am I doing for time? I could talk forever. Let me at least get through cancer. Yeah, you get that sense from me when you're watching me that I love this stuff. And you know, if you guys don't, you know, literally rise up and kill me, I'm going to keep talking. So, cancer. What is cancer? Cancer is the failure of your body to eliminate malignancy. Malignancy will go ahead and essentially grow. It'll blow up like a balloon. It turns out you have lots of defenses in your body that are constantly surveilling for malignancy, and they're eliminating small malignancies, which is why 2/3 of us will make it all the way to a car accident or heart disease or dying of old age before we get a solid tumor. That's not bad. Quite frankly, I mean, that means that your body's natural defenses are taking care of your cancer. It means that everybody in this room has cancer, and it doesn't matter. Because unless it gets ahead of your immune system, you'll never know about it. But one day it might, it escapes. You got 2 cells that are together, then 4, then 8, it's going up like a balloon, and the surveillance now is looking at a very big mass, a lot of surface area, and it can't eliminate it anymore. And that's when you get, what, surgery, radiation, chemotherapy. Well, what we do instead is we take a small amount of virus and put it into the primary tumor. So we find the biggest tumor in your body, and we go ahead and put this virus in there, and it converts about 20% of the cells to secreting something that attacks the natural surveillance cells called gamma delta T cells. So once we put it in there, like I said, about maybe about 20% of the cells will be modified. We start making something called IPP, which is a stimulatory small molecule. It latches onto the microenvironment and it attracts all of these cells that are called gamma delta T cells there, and it activates them and proliferates them. And in an activated state, they eat at 300 to 600 times their normal rate. Of course, they obliterate that tumor, right? So they activate, they turn on the tumor, and boom, gone. But at the same time as they're eating that tumor, they're circulating around your body. And they're in that activated state still. So every tumor that's smaller than that biggest tumor also gets eliminated. The secondary tumors and the metastases. And we've proven this already. In 1,000 mice, we've been able to go ahead and just treat the primary tumor, and all the tumors go away in the mouse's body. 85% of the mice that we treat in this way come back completely cancer-free. Yeah, that's an amazing result. And this really translates to human beings. Why do we always hear that mice are being cured of cancer and yet we haven't found the cure to cancer? It's because of that non-targeted nature. Right? We can't get down to the really powerful things to go ahead and tweak these things. Right? We can't get down to the DNA in the past with these chemotherapeutics. And what's true about a mouse? They're small and they're resilient. We can put a dose of poison into that mouse that he or she can recover from. And if we did the same per kilogram dose on you, you would flatline immediately. Your liver would immediately fail. The mouse's liver can take a pounding, which is why mice keep getting cured, and then it doesn't work in human beings because we cannot use that same dose level. And you know, there you are. It doesn't have enough effect on the cancer. Could clear it in a mouse, but in a human, you can't raise the level up enough. Well, this thing is completely targeted. Right? All it is doing is taking your natural cells, and these gamma delta T cells are so exquisitely specific to malignancy that they've never been seen in science, research, or medicine as ever attacking the wrong cell. They use an NK receptor. And so this should translate beautifully to mice. As a matter of fact, it'll work better, beautifully to humans. It should work better in humans because NK cells, which you probably also heard of, can be marshaled by these gamma delta T cells to raise the effect. So here's the obligatory dead mouse picture. You know that drugs are getting serious when you start seeing a lot of dead mice around. But here's the important thing. This is normal with cancer that we would give to these mice. We put it on their flank. Or we put one tumor on one flank and treat it, and on the other flank we wouldn't treat it. The treatment itself, the vector, does slow down the cancer, but it doesn't do anything without the gamma delta T cells. So we take gamma delta T cells from humans because they're what's called allogeneic. My gamma delta T cells would work in any of you, and vice versa. It doesn't cause an immune response or a host versus graft disease. So we can use the mouse like a human who doesn't have gamma delta T cells. And we can see from this experiment that that's what it needs. Now, if we treat this tumor and they both go away, what does that mean? Yeah, it means that we can create a host-wide reaction. HIV/AIDS, you guys already know, this is old stuff, you know we're curing HIV. If you don't know about that, look at our website, follow us on social media. And what's the future of this thing? A steady drip of these cures, and then eventually a deluge of these cures. And eventually the dam breaks, and all the pharma companies that are up there putting their finger in the dike, buying this product for $1 billion, buying that product for $9 billion, whatever. There's not enough $1 billion payments in the world. But out of the technology, you don't get a handful of new blockbusters or a dozen new blockbusters. You get thousands of cures. There's the revolution. The old way of doing this just won't work anymore. You're going to be curing diseases in your garage one day. A small company is going to be able to get excited about one disease. They're going to license our platform, which has all these tools for curing diseases efficiently. They're going to cure the disease of their choice. And that's what's going to trigger this whole revolution. Thank you.
So we know that the event was going to end about 8 o'clock, but we definitely want to get some Q&A in. If you guys have any questions, feel free to stand up and ask Jeff directly. FYI, the bar's only open until 8, so get your last drink.
Anybody wants to get up and get a drink, feel free. But if you have questions, I'll just hang around for a while, because we don't have to kick you out at 8.
Exactly.
We
So
can keep the building
we'll do
longer.
that, and then we'll have a closing remarks here. So Jeff will be around.